Thank you very much, Carlos, for your presentation, and thank you all for being here this afternoon to share this workshop with us. You will see that I will change completely the topic of compared to the previous talk, because I'm a laboratory person. I will be working all my life in the lab, so I will focus very much on the diagnosis of BRD from the laboratory side. So just a brief outline of the presentation. I will talk a little bit about diagnosis in food production animals, but I will focus very much on the detection and identification of pasteurellaceae, the big family of bacteria that are involved in BRD. I will talk a little bit about the complexity of this group of bacteria, but I will also talk about the new technologies for improving detection and also identification of these bacteria. And finally, I will do a couple of remarks. First of all, I won't go in deep detail into the clinical approach to BRD diagnosis, because the previous speakers already did it. But I want to emphasize that just having a look to the clinical signs in the live animal is not enough. There's always some subjectivity in the observation of the clinical animals, and unless it is supported by a well-designed clinical scoring system, and probably also with the support of the statistics and informatics, there's always subjectivity in this system if it's used alone. And it is basically because there are variability among the different observers, and also variability in the presentation of the disease. The disease could be subclinical or could be severe, so you never know. Fortunately, there are new methodologies and new model systems to follow a group of animals, as a previous speaker already mentioned, and they are basically remote recording systems for physiological parameters, like, for instance, feeding behavior or activity. But still, these methods need more validation in order to reduce false results. So what about the laboratory? The main task of the people like myself working in the lab is testing. So we have to test on clinical samples. And here we have the three areas where testing is necessary. First of all, the assessment of lesions that indicate BRD in an animal. I mean, the necropsy, the investigation of the systems and organs that are involved in BRD. The second testing area is, in fact, our main task in the laboratory, which is the accurate identification of the bacteria or viruses or mycoplasma that are involved in the disease. And finally, some contributing factors that are also that can be also assessed or measured in the laboratory. For instance, the composition of diets or the quality of water, etc., etc. So focusing a little bit more on the assessment of lesions, which is basically a necropsy. When you have a lung coming into the laboratory, it's usually too late. If you have the lung of an animal, it's because the animal is dead. So you already have mortality because of BRD. So that's probably too late. And the second limitation of necropsy is the unspecificity of the lesion that you observe. And that's mainly because probably different agents are or were there and they were interacting. And probably this is just a consequence of the lesions caused by different pathogens in the same animal. And also sometimes we get samples, autolytic samples, because they were not conserved in a proper way. What about identification of the etiological agents, identification of bacteria and viruses and mycoplasma? If you rely your diagnosis on a PCR result, for instance, alone, with no any other data, then the only thing you have is the confirmation of infection. but you don't know anything about disease. This is just a confirmation that the pathogen was there or is in. If you are lucky and you have the tissues and organs, and you can associate those gross lesions to a specific bacteria or virus, then you have more information. You know that you have disease, but you don't know whether the disease was acute disease, peracute, chronic, or just subclinical, if you find the gross lesions at the slaughterhouse. And then, if you are lucky and you have the organs or you have the possibility to make a necropsy, then you also have clinical signs, a good clinical history, then you have a proof of clinical disease. So if this is the scenario, that the task of the diagnostician in the laboratory is, of course, the detection and identification of the etiological agent, and the task of the practitioner in the field is to put all together the results from the laboratory, the clinical signs, and the gross lesions to have a diagnosis in an objective, accurate, and timely way. That's our task altogether. What about the pasteurellaceae? In this previous scenario I show you. Pasteurellaceae, when I was preparing this presentation and I put the list of the pathogens that are generally accepted to be the main causes of BRD, and because I work in the lab with different animal species, we also get samples from pig and poultry, I realized that we have a very similar picture, for instance, in swine. You will also find DNA RNA viruses. You also find bacteria around, and a mycoplasma, in this case Mycoplasma hypopneumonia. And in poultry it's nearly the same. Again, viruses plus bacteria plus mycoplasma, in this case Mycoplasma gallisepticum. So it seems that the respiratory complex is a consequence of rearing hundreds and thousands of animals in a close location. What is common in these three pictures is what I highlighted with the arrows. They are all bacteria, and they are all involved in respiratory disease in food animals. And they are very different among them. I mean, we have here Staphylococcus and also Haemophilus, Pasteurella multocida. They are quite different among them, but there are some biochemical features and some biological features that are common. And that's why these bacteria are all allocated taxonomically in the same family, which is the Pasteurellaceae family, which is a big, big and complex group of bacteria that are range from commensal, opportunistic to important pathogens both for animals and for human beings. Pasteurella multocida is a well-known zoonotic bacteria. They are grouped into 18 different genera and around 70 species. So you can see that the Pasteurellaceae family is complex, big, and very varied. In recent years, there have been significant changes in the taxonomic allocation of the different genus and different species within the family Pasteurellaceae. Here, two examples of the two bacteria that are important for this workshop. The first one, Staphylococcus somni, and the second one, Pasteurella hemolytica. And these were the two first reports of the new name and the new location in the taxonomy list for these two important bacteria, just 10 years ago. So we are just getting used to the new names of these bacteria. And here we have the allocation of these bacteria in the taxonomic scale. And you can see that genus 5 comprises Mannheimia, genus 8 comprises Staphylococcus. With Staphylococcus, no problem. I'm happy with it because it's just one bacteria in this genus. But what about Mannheimia? Mannheimia, as a genus, comprises not just Mannheimia hemolytica, but also other Mannheimia. So that's a problem. That's a problem for me, for people working in the laboratory, because we have to be able to differentiate among them, and it's not easy. This is just to show you an attempt to propose an harmonized profile or a harmonized method to differentiate between different pasteurellaceae bacteria that are important in veterinary medicine, not just ruminants, but also swine and domestic and pet animals, because new data has been introduced in the phylogeny of pasteurellaceae, so we need to refine our diagnostics. But unfortunately, misidentification based just on conventional phenotypical biochemical identification fails, and is common in the laboratory. So it's just an example from the left to the right. We have Mannheimia hemolytica, Mannheimia glucosida, and Mannheimia varegena. And after overnight incubation, these three bacteria look the same. And they can be isolated from the upper respiratory tract of ruminants. So we should be able to differentiate them when we get samples, for instance, a nasal swab from cattle. We should be able to know which of them is the pathogenic one. This is another example. Again, a nice pure culture of Mannheimia hemolytica growing on a blood agar plate. There's blood in this agar. That's why the red color. And this is how the colony growing on top of the blood agar looks like. And if you use a sort of transillumination in this agar culture, you can see here the phenomenon of the hemolysis, the beta hemolysis, which is a characteristic of Mannheimia hemolytica. You can see it as a double ring. The inner ring is the actual colony of the bacteria, and the outer ring is the hemolysis. What is the hemolysis in this case? The hemolysis means that the erythrocytes in the agar plate, in the blood, are destroyed. And are destroyed because the bacteria releases a sort of toxins that destroy the erythrocyte. That's why we see the hemolysis. So the rule of thumb, the paradigm with Mannheimia hemolytica, because let's say the surname of the bacteria, the species is hemolytica. Since beta hemolysis is caused by leukotoxin positive Mannheimia hemolytica, we believe that all beta hemolytic Mannheimia are leukotoxic, and they are not. There are some controversial data published talking about some Mannheimia hemolytica that have the capacity to produce the toxin, but they still show hemolytic behavior in vitro. So that's why some researchers in the US were working with these two phenotypic properties of Mannheimia, the hemolysis and the production of leukotoxin, and they show it as a poster communication in a dedicated conference for pasteurellaceae this year. And they were working, the main aim of the study was to compare both production of leukotoxin and beta hemolysis of a group of Mannheimia. They used 100 isolates, and the results were quite surprising because just 62 up to 100 of the isolates produced leukotoxin, as they detected indirectly using ELISA and other methods. And from the 38 isolates that were not able to produce the toxin, 22 were still hemolytic in the blood agar plate. So that's really surprising. So that's, again, another problem for people in the lab, because not all Mannheimia should be called hemolytica, or they don't produce hemolysis in vitro. What about the Staphylococcus somni? Staphylococcus somni, what is also a paradigm with the Staphylococcus somni is the low sensitivity of the bacterial culture. And it's true. It's very frustrating having no growth of Staphylococcus in an agar plate when we get lungs with the indicative lesions, gross lesions. And the explanation, we don't know. Probably Staphylococcus is susceptible to antibiotics. If there are concomitant antibiotic treatments in those animals, it's difficult to get the isolate. But sometimes we are lucky, and it grows. But it grows very slowly. It takes 2-3 days to show enough colonies. And the colonies are pinpoint. They are very tiny, small. And if you are not familiar with Staphylococcus, then you can miss it easily. And also, the growth of contaminating bacteria usually exceeds overgrowth Staphylococcus. And that happens very often. If we are lucky, again, and we are able to isolate the Staphylococcus and to get a pure culture, then there is no one single phenotypical feature to identify it properly. And even a combination of different strategies give no definite identification. So that's a problem. That's why probably we need new technologies. Now we'll talk a little bit about PCR. And I put a question mark because PCR is not a new technology anymore. It's been in veterinary medicine for diagnosis for more than 2 decades now. But some new technologies are based on this simple principle. Having a tiny amount of DNA, even from a single cell, amplify it, and have enough material to do, for instance, bacterial identification. So several new technologies are based on this simple principle. We know that PCR have several benefits, some limitations as well, particularly with the interpretation of the results, but it still is a tool in the toolbox. Benefits and limitations, just to summarize benefits and limitations of PCR, particularly for BRD. It seems that it depends very much on the origin of the sample and the information that we have in the laboratory or in the field. This is the first situation when we have the sample from the nasal, from a nasal swab, from the upper respiratory tract. If we do a PCR, the only thing we are doing is detecting the DNA from the bacteria. But it doesn't give any clue about the disease. So we just know that the pathogen was there. But we cannot differentiate between just incidental infection or the inspiration of droplets of aerosols with the bacteria. We don't know. If we take the sample from the lung, then we could eventually associate the identity of the bacteria or virus with the lesions. So we have more information, but still we cannot be sure that the bacteria that I identified is the real causative agent of the disease. PCR and BRDC. PCR is used for BRDC diagnosis on routine basis. This is a recent paper dealing with a comparative study of PCR and the conventional culturing. Very interesting because they just take some samples and they compare positives from PCR, positive from the culture. They worked with 150 pneumonic lungs collected in 2013, and they did the standard culture and PCR on the same sample. And the results are summarized in this table, but the conclusion is that there was no excellent or perfect correlation between culturing and PCR. Some of the culture negative were PCR positive. That's quite a common scenario in the laboratory, and probably is the main criticism against the PCR particularly for Staphylococcus somni, for instance. But also they had positive cultures with negative PCR. So there was something wrong with the PCR in this case. So again, there was no perfect method to detect the bacteria that are involved in BRD. So just to summarize the use of PCR in BRD, we can use PCR to identify bacteria like Mannheimia hemolytica if we have a species-specific PCR. And we can also use it to improve the identification of Histophilus in cases of BRD. Another application of the PCR is the semi-quantitative PCR. This is how a modern semi-quantitative PCR looks like in the screen of the computer. Here we have an experiment where we use bacteria, and we try it with 10-fold serial of a specific bacteria. Then from the left to the right, we have different PCR signals. They were all positive, but some samples showed a positive signal before in time. It's semi-quantitative. This PCR is semi-quantitative because we can count something. Not just say it's positive or it's negative in the PCR method, but we can count. We count time, because PCR is a dynamic process that occurs in a period of time. And we can count here cycles of amplification of the DNA. Usually they go from 0 to 40, is more or less a standard everywhere. So here we have samples that were positive because they produce a sigmoidal curve, that means positive PCR, but at different time points. Some of the samples were positive before, and some of them were positive later. And the higher the amount of DNA, the earlier the signal was detected by the machine. So in this way, we can establish categories of PCR. The higher PCR signals, I mean, those samples containing more bacteria gave a signal before and stronger. And if we use the same principle than in bacteriology, when you have a blood agar plate, for instance, plenty of mainly Mannheimia hemolytica, then you establish a relationship of cause and effect. So we can use the same principle for quantitative PCR. We could say that probably the first sample contains a lot of DNA from, let's say, Mannheimia, so probably Mannheimia was involved in the disease or in the gross lesions. So this is the way how we interpret qPCR results or quantitative PCR results using a scoring system, taking into account the amount of DNA that we find in the samples, high, moderate, or low. And we just use a simple scoring system to quantify DNA on samples. Multiplex PCR is another variation of PCR, and it's based on the same principle of PCR. Just the amplification of DNA from an agent, but we can identify several pathogens in the same PCR run. So there's basically the same principle, but with the modern machines and with the modern reagents for PCR, we can do this. So why multiplex PCR for BRD diagnosis? Because BRD is a polymicrobial disease. So several agents, several pathogens can be present in a lung at the same time, same animal. So this is a recent paper where, dealing with synergism or combination of a virus and a bacteria that are involved in BRD. BRSV and Histophilus causing an enhanced gross lesion or microscopic lesion, in this case in a lung. So if we are in this scenario, probably it's a good idea to have a multiplex PCR to detect both the bacteria and the virus. And you can have a look to the scientific literature, and you will find different studies dealing with the use of multiplex PCR to detect several pathogens from BRD cases, particularly for viruses. Finally, I will talk about the MALDI-TOF technology, which is a new technology. It is not based on DNA or whatsoever. It's a totally different approach, and I will try to explain you the principle. The actual bacteria, the test bacteria that I don't know its identity, is fixed in a sort of metallic plate within the machine. And then it's covered with a matrix, it's a sort of synthetic material, and it's fixed there. And then it is irradiated using a laser beam. And the bacteria is in some ways is destroyed and is broken into molecules. And these molecules travel along the tube of the machine where they are ionized. And then there is a detection system that is able to detect the different molecules because they have different characteristics. Some are bigger, some are more complex, some are smaller. And they produce, the machine produces a sort of a spectra, which is like a fingerprint and is unique for each individual bacterial species. So in that way, we have a very nice spectra for each individual species of bacteria. And why I mention MALDI-TOF? Because MALDI-TOF is becoming more and more popular, mainly because it's a fast technology. It's even cheaper than some DNA-based technologies like sequencing, and because of that, it's becoming more and more popular, and it's used now for species, bacterial species identification. And what about MALDI-TOF and pasteurellaceae? Here's a report of some researchers in the US, in Northern Ireland, sorry, working with 250 strains of pasteurellaceae comprising different genera, different genus, and different bacterial species. They use reference spectra. You need a reference spectra, of course, to compare your test bacteria with the known one. And the results were quite good, but still not perfect. For few bacterial species, there was not enough discrimination power. That happened mainly because they had at that time, enough reference spectra to compare with. And still the equipment is expensive, but more and more private laboratories are acquiring the equipment and are offering the service to external, to third parties, to other laboratories. And the last paper I want to show you is a comparison between different approaches for bacterial identification. It's a DNA-based, the first one. The second one is a semi-automated bacterial identification system, which is the conventional system. And finally, the MALDI-TOF. And again, the results were not perfect. Still, some bacteria were not identified. But the most interesting conclusion from this paper is that it highlighted the contribution made by people in the lab. I was very happy when I read this conclusion, because we should know the basics and the fundamental behavior of the bacteria in the lab. And we should know if it's gram-negative or gram-positive, or more or less the family the bacteria belongs to. Finally, a couple of remarks. The multifactorial and polymicrobial nature of BRD requires a comprehensive teamwork between the practitioner and the diagnostician to get a common target, which is an accurate diagnosis of BRD. And finally, the new technologies are and will be of great help to improve the conventional diagnosis, particularly bacteria and BRD. But still, some of them requires more validation, and some of them are still under research, but for sure in the future they will help a lot. Thank you very much.